All right, Noah, we're live here. Just wanted to make another recap video going over what we talked about tonight in the review session. Most of what we focused on was the slides here about the overview of neuroplasticity, the ability of the nervous system, particularly the brain, to change its structure in response to experience. And so slide one is just talking about, this is just also for me to help review and go over about how the neurons communicate, and that's like the synaptic transmission stuff we looked at in the last slide. So electrochemical flow, it calls action potential movement down the axon of the presynaptic membrane. That's increased its speed. As in one thing we focused on tonight was talking about how glial cells, it's oglamidopteron, blah blah. I'll pull up the spelling, but one starts with an O. It's a longer word. That's the one that's part of the central nervous system, and then Schwann cells are part of the peripheral nervous system. Both create myelination, which is one of the slides in here, that increase the speed of transmission because they create, I don't know why, but they wrap around, and then the gaps are called nodes of Ranvier or whatever. And I think you probably remember that. So that's pretty basic review. So slide two, talking about turning on gene expression in the neuron's nucleus, causes production of proteins. I have enzymes here, cause chemical reactions, which make more enzymes. Synaptogenesis, creating connections among the neurons. So we have a exuberant synaptogenesis is when like there's a young baby, early brain development. That's exuberant synaptogenesis. Also includes strengthening connections among the neurons. So this right here is a GIF in class of it connecting. So think baby, brand new connections, strengthening of those connections among neurons. We talked about athletics as an example of this, creating mind and muscle memory. She also mentioned, this isn't, I don't think it'd be on the exam, but she talked about how, people who learn to play the piano and move their fingers really fast. That doesn't come from their brain processing each individual one, because their brain can't process that quickly, but rather it's just, that learned muscle memory. So synaptogenesis, the creating of those new connections between different neurons. So other forms of neuroplasticity, so that's just like your natural genetic expression is like, boom, new connections, life forming, and then strengthening those. And so we talk about like there's the prime learning age when you're younger. So myelogenesis, circuits are faster and more efficient when, there's that long word I was looking for, oligodendrocytes, the glial cells in the central nervous system, right here, oligodendrocytes. Produce myelin. Myelin we talked about are associated with many hours of neuron firing. So I guess that's the use it or lose it idea with your neural connections. Makes action potential 100 times faster. Overall enhanced function 3,000 that of unmyelinated fibers. So myelination 3,000 times enhanced, 100 times faster. Action potential 100 times faster. There's only one question where she said you might need to know a specific number, and I'll review that when we get there. Where did B go? All right, so B is neurogenesis, differentiation of fully mature neurons from stem cells. New neurons are continuously. We didn't really talk about this. I should have asked more questions. I might post something on the discussion board. New neurons continuously generate a precursor called the cell lines, the ventricles, migrate to the neocortex. New neurons added selectively to association of. So I guess all we need to know, we didn't really talk about it, so I don't think it'll be that important. She'll probably grab at least one question from each slide. So just knowing that it's not enough to repair the CNS. I mean, that's probably also in your notes. I don't really have anything helpful to add there, so we'll move on to the next one. Just know that it can't repair CNS. That's more for myself, and that it's you can create new neurons, but it's not that much. All right, epigenesis. The environment around neurons alters control molecules that regulate expression of genes. We talked a pretty decent amount about this. I think this is where we opened our discussion. I'm trying to look at my notes from tonight, because that's what I want to focus on. I know you have the other notes. I want to focus on that. But we talked a lot about the mice, and then we talked about how children in depraved environments. That might be something else, though. She did talk about that, though, from the other points to ponder, or in earlier lectures, not part of these slides. really important to know is that children in depraved environments, so depraved of stimulus, so you think about that cat example with the eye sewn shut and how the neurons didn't, what's it called? That neural connection wasn't made because of the lack of stimulus. So we looked at children who were deprived of stimulus at younger, developed thinner dendritic spines, which means I think they had less myelination. We looked at that chart of like the white brain, how it became white because of the myelination. So it's less efficient. So we can look at, let me just keep cranking through these sides. The experience does not alter the DNA sequence. So DNA does not change. She talked a lot about true-false questions. That'll probably be related to this. Let me see if I can write those down. There's another key thing we need to talk about too. Long-term inserting neurogenesis. Yes. Oh wait, here's another. I just have neurogenesis creates new neurons, plastic yawn, synaptogenesis, epigenesis. Nothing really on epigenesis. Maybe. Synaptogenesis. Epigenesis. Here it is. Epigenesis. Most life. Okay, so that's what we're talking about. She talked about also this example with rats for slide C, epigenesis. That they looked at rats who overate, and then that it made the kids more likely to overate. But a lot of true false here. So just remembering that. It does not change the DNA, but rather the regulatory molecules that affect DNA expression. So like the scent one too, the scent in the mice, how the mice were like exposed to that smell that was like associated with danger, and then later generations had that same thing. So pass through parents, grandparents, sperm and egg. Cool. Slide D, probably gonna need to know all those. I think you have them written down. I'll just run through them really quick. Aerobic exercise, good sleep, consolidate learning from the day, especially REM sleep. Good nutrition, relationships, novelty. I don't know what novelty means. The close paying of attention, avoid multitasking distractions, care what we are focusing on, time in when we reflect inwardly, possibly humor, preliminary studies on laughing. Okay, I don't really know what that meant, but that supports neuroplasticity, the brain's ability to mature and changes neural connections, I guess. All right, experience-dependent synaptic plasticity in the brain. All right, we talked probably a good, of the hour we were together, we probably talked a good 30 minutes on this. Probably going over slides under this section, going over C through F and G. And before we get too far ahead of ourselves, let me go through my notes and see what else I have written down. So we thought methodology is something key. There's slides in our lecture that we don't have in our notes. She talked about methodology, so like ways we can measure the electrical impulses within a neuron or within a cell. I don't really know exactly. And we talk about how we can measure an electrode. What would be a means of gathering resting potentials of neurotransmitters? Maybe I was right. So what would be a means? The presynaptic. So if we want to measure the resting potentials, there's a thing called an oscilloscope. Neurons action resting potential. Oscilloscope. It's not in our slides, but it's like this little machine that measures millivolts, and you can insert it. So oscilloscope, spelled oscilloscope. That's approximation. We don't know actually how it's spelled. Science doesn't know. I'm sure you can look it up. Okay, so that's one of them. Electrochemical flow, movement of the neurotransmitters. Calcium binds to vesicles. Oh, we talked to one of the kids, that Joey kid from Wheaton, asked about what's it called, or went to St. Francis. He asked about the calcium process and synaptic transmission that we talked about last time. So the neurotransmitters start in the vesicles. So we have that presynaptic, call that the male end, the female end, postsynaptic, and then those little vesicles have the neurotransmitter in there. The calcium comes in, opens them up, allows them to flow out. Those are voltage-gated calcium channels that allow the release of the neurotransmitters. We talked about the autism being, if you're define, what is autism? Define it. It's a developmental disorder, often generalized by too many neural connections within the brain. Autistic brains tend to be much larger. And And so there's so much interaction and firing within the brain that hasn't matured, that there's confusion with all the signals. And then that first sentence on the point to ponder slide will explain what that kind of looks like. All right, so that's that. We talked about epigenesis. So those are the switches that turn off and on the genes that are expressed. DNA is the same, but epigenesis suggests that the switches that turn on features will go on and off. And those changes that turn them on and off are inheritable. So the genes involved in epigenesis do not change. That's correct. Okay, so that'd be like a true-false question. The genes involved in epigenesis do not change. That is true. They do not change. Genes do not change. DNA stays the same. Nodes of Ranvier, oligodendrocytes. Oligodendrocytes, that was on that first slide. Going over that, which is in the central nervous system, they produce myelin. Schwann cells are peripheral, also glial cells produce myelin. Okay, she talked a lot about methodology. She says, because it's a SIP class, you must know this information. But we're like, we don't even have this in our slides. And she was like, I wish somebody would have told me that. But I think she's still going to ask questions about that. So here's what you need to know about methodology on the stuff we don't have slides on. She talked about tonight. I hope this is important and helpful. I don't know how much time I have left. Okay, so one methodology that how science measures neuroregression. So once we move into the talk about mechanisms of synaptic plasticity. So we looked at synaptic transmission last time. So this is how that changes. What is the neurons this is the wiring of neurons that fire together or wire together. This is how that wiring occurs. So once that sensory thing is fired, how do they wire together? So we're looking at how and then if they are wired together, how do they degrade? So we're kind of on the latter half of that. So moving through slides F into slide G, we're talking about long-term synaptic depression. That is where they fire out of sync, they lose their link, so they're no longer, those connections aren't as strong. So how do we measure neural regression? And so there's two ways. One is intercellular recordings, and the other one is immuno, IMMUNO, histochemistry. Chemistry? I don't know. Immunohistochemistry. It's like immune history chemistry. So all those. So intercellular recordings, immunohistochemistry. Intercellular recordings can be used to detect the number of synaptic connections. So I wish I had those slides. Oh, those are the ones where we counted out the numbers. It was like 5 to 1. That maturing process. So as we mature, that's why like the autistic brains, like actually right here, we can see, we get those less connections. We talked a lot about that. Or even this is an example. So this is like a zoomed out version of what's happening. So we have A and B. A now has less connections. It got used less. So those connections weren't as strong. B created more. What we're doing is looking at a zoomed in version. What creates those more connections? And what we talked about is, this is all classified under the CAT thing. So the Heb, these are all Hebbian modifications. So these are all versions of that. He didn't know that all this was going on, but he was the first to discover that this process could even occur. So this is looking at a zoomed in version, the mechanisms of synaptic plasticity, how this happens. And so I want to tell you again about these two methodologies. Intercellular recordings, measuring the different amplitudes. And so we measure, that's within a specific cell. So intercellular recordings can be used to detect the number of synaptic connections, regression of synapse through maturing. So in theory, it'll go down. And then, so it measures within a specific cell. Immunohistochemistry, it counts the number of synapses using the coloring of the neuron. So we'll look at a neuron, and I wish I had a picture of it. Maybe if I Google it, I can still show you on here. I want to go fast, though, because I know we're going to run out of time. Immunohistochemistry, boom. We'll just click on images and see if it pops up. No, this looks like that's helpful. Oh, this is kind of what it is. So it has like green and blue. I don't know. We looked at like a more in-depth version, and it was like a process, but essentially they become one color, and it means that it's maturing, I think, or one is going away, essentially. So here's a key number she wants us to know. Regression in the normal person. So as a normal person, as they develop, how much of their neural, so they're not autistic, basically, how how much they lose. I don't know what slide this was on. It's on one of them. It's on... I don't know where it's at. It's in one of the slides. It talks about the normal